All right, so we are going to give a quick try to a session on YouTube to review for the AP exam. So we are going today to do a short review on sensation and perception. So I'm trying to run this off of both Instagram Live and on my YouTube channel. So then the YouTube channel one will be saved. So what I'm going to do is just go through some of the key phrases, some of the things that you definitely want to know about sensation and perception. And we're going to take a look at a couple of different things to make sure that we try to demystify a little bit with sensation and perception and make sure that you are ready for the exam. All right. So let's see, trying to figure out what the best, there we go. So I'm going to talk a little bit about some of the key terms. You will start with sensation and recognizing that you need to know the difference between sensation and perception. That sensation is the process of getting all that information in. All of the things that you experience in your environment that come from a stimuli energy. So we're including the eye, the ear, we're including the sense of sight, the sense of hearing, the sense of taste, which is also called the gustatory sense, the sense of smell, which is the olfactory sense, as well as the sense of touch, kinesthetics, and your vestibular sense, which has to do with your sense of balance. So make sure that we are familiar with some of the key topics being sensory transduction, looking at absolute threshold, looking at the difference threshold, looking at the different theories for each of those. So as we begin to take a look at that process, let's start with getting information in. This is bringing the information from the environment into your brain through your senses. So what has to happen is light, sound, all have to make it into your brain to be processed. So what we're seeing here is top down, bottom up. So the process of sensation is really about the bottom up. It's getting everything from your environment into your brain to be interpreted and given meaning. That is the bottom up. Top down is when we are applying meaning to it, interpreting it, converting all of that information, all those sensory stimuli into a message that your brain can understand. So that's the top down part where we're giving it meaning and interpreting it. The idea is signal detection theory is one of our basic theories that tells us that we are interpreting our environment on the basis of selecting which pieces need to be attended to. Signal detection theory gives us this idea that it's 50% of the time. When we identify a stimuli 50% of the time or more, then it crosses our absolute threshold. That absolute threshold is when we can then identify it accurately as a stimuli. Otherwise, think about the number of sights, sounds, smells, etc., that exist around us all the time. And it'd be way too much to process. We wouldn't be able to interpret all of that information. It would be so crazy overwhelming that our brains couldn't handle it. So instead, we filter it. We take the information that we really need to know. For example, if you're standing in your house, I happen to be standing in my kitchen right now. See, there's my feet. Are lucky you. Standing in my kitchen and doing a little bit of talking. And if I were to constantly process the pressure that I feel as I'm standing on the floor, my brain wouldn't be able to process other information. I wouldn't be able to pay attention to the level of light. I wouldn't be able to pay attention to the sound of the kids in the basement playing. So our brains need to be able to attend to the things that really matter. So we filter out a lot of it. So that signal detection theory helps identify the fact that stimuli need to be present enough for us to notice them. And to notice them, you only have to notice it 50% of the time. So that first identification then brings us that information. And then what can happen is as we move around and stand, we can begin to experience what's called sensory adaptation. And that sensory adaptation is the fact that we get used to that stimuli, and the continued presence of that stimuli diminishes our sensitivity to it. So as you stand on the floor for an extended period of time, and that extended period doesn't even take that long because the pressure isn't that great when you're just standing, you're going to feel it less and less. Here's another example for you. Think about the last time you cooked something that has a strong odor in your kitchen, whether it's coffee, cookies, fish, onions. When you first cook it, you smell the smell of that food. It's overwhelming. By the time you go to eat it, even though you're still smelling it, the intensity has significantly diminished. And that's not because the number of molecules of the food item are in the air around you, but rather the fact that we've adapted to those scents. So think about if you wear a watch or you wear any jewelry over the course of the day, you don't notice the presence of that item anymore. Think about hats and glasses. Those are really good examples too. And the fact that we continue to, it doesn't change the amount of pressure that is on your skin by wearing the watch all day. So why do you only notice it when you first put it on? That's because of sensory adaptation. So then let's take a look at what happens as we continue to experience these stimuli. How do we then notice the difference between something that is a light pressure and something that's a hard pressure? Something that feels warm versus something that feels hot? How do we notice the difference between something that's very light to pick up and something that's a heavier version of it? Well, this is where Weber's law and comes into play. And what it says is that things vary by a constant amount in order for us to notice the difference. This is also called the just noticeable difference. So this applies to our perception, to our experience of any of our senses. So whether we're talking about the amount of light in a room or the volume or the pitch of a sound or the intensity of a color, could be anything. It has to vary by a set percentage. This is Weber's law in order for us to notice the difference. So if you're listening to a sound as the tone increases in volume, it has to increase by a ratio, by a certain percentage, not by set number of decibels, but by a ratio. This again is Weber's law. Okay, so that gets us through the initial feeling of sensation, getting it in, understanding the process of the just noticeable difference. Make sure that you understand that you remember the term transduction. Transduction refers to the translation, to the conversion of sensory stimuli, turns one form of energy into another. So it takes our environmental stimuli and makes it into brain language, something that our brain can understand. So that's a term that we're going to see in each of the different senses. You want to make sure that you are familiar with transduction because it occurs in each of the senses. So think about a couple of different terms also that are associated with sight and with sound. You're going to be thinking about wavelength and intensity, frequency and amplitude, looking at the fact that wavelength refers to the distance from the peak of one wave to the distance of the next, where intensity is about the amount of energy, and this is measured by amplitude. So when we are looking at the eye, hue comes in the basic colors of red, green, or blue. And you want to make sure that you think about the concept of the visible spectrum. The visible spectrum is pretty small for humans. Take a look. I'm showing you this beautiful thing out of a textbook that I have. It's a different book, a prep book for the exam there. Can you see that? This shows us the visible spectrum of light. You've got your long radio waves up here and your gamma rays down here, neither of which we can see. Here are your X-rays and your UV rays, your IR, your microwave, and your radio waves. Look at all of those waves that we cannot see. And this right here, this teeny tiny little line is the only one that we can see. So what's important really is just to remember that we have a very small spectrum of visible light. So let's take a look at the parts of the eye. The eye, let's see, turn some lights on here. See, there we go. Here is the drawing of the eye, and a term that we have not used in our textbook, in the Myers textbook, is this vitreous humor. The vitreous humor refers to that it's filled with it's where the iris resides. The iris is in that chamber called the vitreous humor, and it holds the aqueous humor is filled with a watery fluid. So what happens is it comes through the aqueous humor into the vitreous humor before hitting the retina at the back. Now remember, when you get the image coming into your eye, it's actually inverted on your retina. So here is the outer part of your eye. Remember, you've got that cornea protecting your pupil and your iris. And remember, the iris is that colored ring that is muscle that controls the opening and closing of the pupil, which controls how much light enters into the eye so that we can focus our images. So what's happening here then is your lens is going to, remember the term accommodation, shift in its thickness or thinness and in its how oval it is. It stretches and shrinks so that then what happens is it's focusing the image right here on the back of your retina in this central area called the fovea. The fovea is where we have the highest, most clear in focus image. This is where it's perfect, where we can see exactly what we are looking at. So this back part is your retina. This is where all of your rods and cones are. Remember your bipolar cells and your ganglion cells. Remember the ganglion cells are over here. Your bipolar cells connect between the retina and the rods and the cones, which kind of makes sense because guess what? Bipolar means two sides. So they connect here and talk between the ganglion cells of the retina and the cones and rods that are also residing at the back of the retina, and the bipolar cells helps make that connection. So the image comes in across the cornea, through the light passes through the aqueous humor and then on through into the vitreous humor. Keep those off to the side in your mind. So it comes through the cornea into the lens, where the lens then is going to focus the image. Now, at the same time, the iris and the pupil, the iris is controlling the opening of the pupil in order to determine how much light is needed to enable the lens to focus the image. Then the image will be focused onto the back of your retina, where it is then passed by the bipolar cells into the rods and activating the rods and the cones, and passed over to the optic nerve, where the process of transduction occurs. So the optic nerve then carries this information into your visual cortex. So here's a great little chart. Great little chart that shows you the process, the steps of vision. So you go from the light waves, pass through the cornea to the pupil, to the lens, to the vitreous humor, to the retina, where you've got your rods and cones. Your bipolars and your ganglions are then going to assist in the process of taking that image over onto the optic nerve. The optic nerve, remember, where do all of our senses go? You got it, the thalamus. So the thalamus then is our sensory switchboard and is going to relay the information over to the visual cortex, where the information is then processed and then sent back out to be interpreted and dealt with. So for example, you don't walk into a wall. So you can see there's an awful lot of steps going on in order for us to see something. So that's part of the process there with sensation. And think about the fact that is just one piece. So here we go into the rods and the cones. The rods and the cones are located at the back of the retina. The rods and the cones um, are going to be responsible for our peripheral vision and for our color. The rods are responsible for looking primarily at the light conditions. When it's in a dark room, more of your rods are activated. And then we need less of them in well lit conditions. So these rods then help us also with peripheral vision. The rods and the cones work together, and the cones are responsible for our color vision. So there are a couple different theories as to how we see cones. Cones are for, as I said, the color. Remember the C and the C for color vision. So the cones, there are several different theories as to how we actually see color. And I know you're not really seeing much color right now because you're looking at the image of the eye from our packet, which is in black and white. Anyway, let's talk about how we see color. Several different theories. Number one, the trichromatic theory. The trichromatic theory says that we have three cones: red, blue, and green, and that every color that we perceive is actually a mixture of levels of stimulation of those three different color cones. Each cone would be highly specialized, and these are located, as I said, in the eye at the back of the retina. Other theories say that it's really not about the trichromatic theory, that it's really more something that's occurring in the brain and in our interpretation of the information that's being sent in. So this is called the opponent process theory. The opponent process theory says that we have these different cones, but that it's really more about how color activates the different pairings of cones, that all of our cones are paired off permanently. So you have red and green, yellow and blue, and black and white. And those are, so if one of the colors is on in that cone, the other has to be off. So you can only either be perceiving at one time with only one cone, which, remember, we have thousands of cones. One cone at one time is only either able to see red or green. It's not able to process both. So looking at the opponent process theory, this helps us to understand why, when we look at the red and blue flag for an extended period of time without blinking, and then we look over to a white piece of paper and focus at the black dot in the center, you'll see the reverse images, which are the green and the black, because we have looked at the red and the blue. Then we'll see the opposites of those colors and see the green and the yellow as opposed to the red and the blue, because your tired eyes get your eyes get tired from watching that same color for a period. So we switch over. The concept of an after image is the fact that we do continue to look at something, and particularly if you're not blinking a lot, when you look away, the residue, that little bit of image that you still perceive, even though you're not actually looking at that image anymore, that's called an after image. Okay, so unless there are any questions about the eye, those are the big things you need to know about the eye. Remember, the visual cortex is located in the occipital lobe here at the back of the brain. If you were to put your hand right onto the back of your head, you'd really hit it. So here's what happened, which is why when you fall off your bike and smack your head, you see black. Your field of vision has been interrupted. So if you imagine that these two circles are your eyes, we see the images coming in. Remember, your left eye is connected to your right side of the brain. Your right eye is connected to the left side, goes in through the eyes, across, transduced onto the optic nerve, through the thalamus, into then to be processed by the visual cortex once it has been routed appropriately by the thalamus, and then we can make sense of that image. Okay, remember that parallel processing is a term that means we are able to interpret several different things at once. For example, you can look at this image and see both the changing coloring reflecting, the changing light reflecting, and the fact that it is moving locations on you. Isn't that nice? Yeah. So parallel processing is the fact that we can see things and perceive several different things at once. So you can't really see anything. There's no airplanes going by or dogs out back. But if you can imagine a dog running across the yard, the fact that you could identify the shape, the size, the color, and what that must mean that the creature is, if it was a dog running across your yard, then that's an example of parallel processing. So that takes us on. Let's talk a little bit about color blindness. Color blindness, just as a really short side note, is the inability to perceive color. And it's really not about only being able to see black and white. It's caused by a lack of particular types of cones. It is genetic. It is very heavily male as opposed to female. Most people experience what's actually more accurately called color weakness than color blindness. True color blindness is extremely rare. Red green is the most common color deficiency, and it is absolutely a recessive trait on the X chromosome. We do see it in more men than women. So that takes us on to the ear. I'm not seeing any particular questions. So let's go ahead and talk a little bit about the ear and theories of hearing. So make sure you are familiar with, here, let's start with audition. The whole process of hearing is called audition. This is the sense that we call hearing. Sound waves are created, and how we interpret them is the whole process of audition. So amplitude affects the quality of what we consider loudness, sound pressure, or intensity, and the wavelength affects the pitch. The sound's frequency is the number of wavelength cycles in a set unit of time, and they are measured using hertz. 1 hertz is equal to 1 vibration per second, not something that you're likely to need to know for the exam. Let's talk a little bit about the physical structure of the ear, the anatomy of the ear. The outer ear, that funky shaped outer ear, is called the pinna, Pinna. The pinna, this part, your outer ear piece. It has a design that allows it to catch the sound waves and to direct them and carry them into the auditory canal. The auditory canal then takes the sound waves and vibrates the tympanic membrane. This is what we call conduction. The series of vibrations is what then through this auditory canal, it vibrates here on the eardrum, which stimulates the oval window. So then what we see is, if you, for example, are having trouble hearing something, we typically will turn our head towards the source of the sound. The reason we do that is to help catch more of the sound waves into the center part of your pinna, so that it can focus the sound waves into your auditory canal a little bit better for you. So yeah, it actually does help when people cup their ears and begin to turn their heads towards the source of the sound, because you do collect just a little bit more of those sound waves. So the bones are also those, when the sound waves come through here, they stimulate the oval window. The tympanic membrane here is this eardrum is going to then allow the message to carry over into these bones of the middle ear. These bones are also called the auditory ossicles. They are very, very, very hard to break. They are called the hammer, the anvil, and the stirrup. Remember, the hammer, the anvil, and the stirrup are the smallest bones in your body, and they are really strong, probably somewhat because of their size. They concentrate the vibrations of the eardrum onto the oval window. So here we go. Here's our sound waves coming in, stimulating here, passing on to the hammer, the anvil, and the stirrup, which are focusing the sound here at the oval window. Now, that's going to be an important place for you to remember, because the oval window is then going to be part of our theory of pitch, how we sense pitch. So the sound comes through from your pinna into the auditory canal, stimulates here the eardrum, which then causes the hammer, the anvil, and the stirrup to vibrate, passes the information here, then accesses it, stimulates the oval window. Wow, it's loud in my house. So once this has happened, the oval window, the inner ear, we now are moving into what we call the inner ear. This part here is the middle ear. This part here, the cochlea, is our inner ear. And let's talk a little bit about what's happening now. So the hammer, the anvil, and the stirrup concentrate the sound here on the oval window, pass it into the cochlea. The cochlea carries the sound here. This is a fluid-filled chamber, and this has a lot of little, it's the inner surface of the cochlea is the basilar membrane, and it resonates to different sounds at different locations. On the top of this is the organ of Corti, which contains hair cells. We don't need to know about that. So then here's what happens. It's carrying the sound around through the cochlea. The fluid, here's the direction of the fluid, so it's constantly moving, which explains why when you start spinning around and then stop, it makes you feel dizzy. The fluid continues to move. All right, so this fluid-filled chamber, the cochlea, is going to be carrying the sound over here to your nerve fibers, your auditory nerve, where it is then going to be converted into a message that the brain can understand. So, sound waves through the pinna into the auditory canal, where it then carries it to the eardrum. The eardrum is stimulated, where sound then passes through and moves on from the eardrum to the hammer, the anvil, and the stirrup. These little three bones then stimulate the oval window, carry the sound through the cochlea, and then carry the information here over to these nerve fibers that will then take it to the auditory nerve. The auditory nerve is going to transduce the message, turn it into something the brain can understand, carry it through the thalamus, and then over to the auditory cortex. So this auditory nerve is what is very heavily responsible for converting the sound, excuse me. All right, so here's another image that you might like. This particular one shows you the organ of Corti. I'd be very surprised to see that in there. You can see it's where we've got here the hair cells and the basilar membrane that then protect this little area, the organ of Corti. This has to do with the auditory nerve and the process of transduction. So let's talk a little bit about the different theories of sound. Sound vibrations give us both amplitude and frequency. Hearing is when you're looking at the process, how do we hear pitch? It could be the possibility of what we call place theory. Place theory says that it's all about where on the oval window is stimulated, the place on the oval window that is stimulated by the passage of the messages that are being received. Um, we've also got another theory that is called frequency theory. So you've got place theory, where on the oval window is stimulated, frequency theory, how often is it being stimulated? And that process differentiates the two possibilities. That shows us the different two possibilities of how we hear pitch. Okay, so several different issues with loss of hearing. Conductive hearing loss versus sensory neural hearing loss. Conduction hearing loss occurs when there's poor transfer of sound. It is conducting or lack thereof. That's why it's conductive hearing loss. So, conduction hearing loss is the most common, and it is something that happens to most people as they get older. This is what is helped by hearing aids. Conduction hearing loss can be accommodated for by using hearing aids. Unfortunately, these are absolutely worthless when it comes to nerve deafness or sensory neural hearing loss because of the fact that they don't have something working properly, whether it is the cochlea or a piece surrounding that works with the cochlea. It doesn't matter. But if that's what's not working, hearing aids are not going to help. Sensory neural hearing loss is caused by damage to the inner ear, typically the cochlea or the hair cells. And what this does is it prevents your auditory cortex from processing the information because it doesn't get there appropriately or quickly. 90% of sensory neural hearing loss is preventable. So, ladies and gentlemen, turn down the volume just a little bit. It doesn't have to be exceptionally quiet, but it is thought now that extended exposure to loud noises does have the ability to cause long-term hearing damage. Okay, let's talk a little bit then about the sense of touch. The sense of touch is based on nerve endings that are in our skin, and the sense of touch is also called the kinesthetic sense. So, the sense of touch is responsive. What it does is it receives signals from receptors, and then they're interpreted by the thalamus and sent on to different parts of the cortex. The more receptors there are in a given body area, the more there is devoted in the brain to it. So, what we notice is that particularly sensitive areas of our body have a significantly larger piece of the brain that is dedicated to interpreting that message. Okay, so in terms of the sense of touch, we've also got gate control theory that doesn't really fall within touch, but it is part of its own. Gate control theory deals with the process of pain. What it says is that there are basically two different types of neural fibers that are stimulated, and those fibers, when they are stimulated, so that we perceive something painful, it's uncomfortable. And the reason that it becomes available to even be stimulated is that the extended or extensive stimulation of neural cells on the basis of quite often touch. Okay. So kinesthesis is our sense of movement. You want to make sure that you understand that kinesthesis has to do with our awareness of where our body is and where our different parts are and how they might respond. One if you think about the vestibular sense is very tightly connected to your sense of hearing. The vestibular sense is about our sense of balance. It is associated, for example, with motion sickness. Sensory conflict theory says that it is actually a mismatch between information from the body and the vestibular system and kinesthesis, not the cochlear fluid-filled sac that is responsible, for example, for dizziness. So that takes us on. We've already talked briefly about the sense of taste also being called gustation. The gustatory sense gives us four basic sensations that are on our tongue with those taste buds. What we see are sweet, salty, sour, and bitter. And now we are also saying there is umami. This is savory or meat. Our sense of smell is also referred to as our olfactory sense. The sense of smell gets to go from the olfactory bulb directly to both the amygdala and to the hippocampus. The sense of smell does not have to take a trip to the thalamus every time it goes to the brain, which helps us understand why the sense of smell is so strongly related to memory. All right. So our sense of smell, what we get is sensory stimuli into the nose where it then stimulates those hairs on the inside of your nose are filtering out some of the different dusts and dirts from our environment, carries it in. Eventually what's going to happen is it's going to wind up on that olfactory nerve and go through the olfactory bulb. And what that then does is gives us an interpretation of that smell. Chemical receptors go to the olfactory bulb, to the amygdala, to the hippocampus. Okay. So this takes us to some interpretation of some different sensation and perception issues, particularly the sensation ones. Make sure that you remember absolute threshold, difference threshold, and Weber's law. All right, so that's going to take us over here to the issues with visual perception. Some of the things that you want to know are the different types of psychology, making sure you remember that Gestalt psychology has to do with a group of people who were particularly interested in the fact that the sum of the parts are significantly greater than the one measurement. So in other words, Gestalt psychologists were all about a complete image or a complete sound, or because ultimately in the process of sensation and then perception, we experience things, experience the world as a whole unit that gives us significantly more information than only getting a little bit of it. All right. So that takes us to the principles in Gestalt psychology. So the principles with Gestalt psychology are proximity, similarity, continuity, and closure. Proximity, the fact that things are closer together, we tend to interpret them as belonging together. So, for example, when you take a look at this review sheet, the fact that these letters are closer together helps you to see that they must be a word. It must be forming together to look at a word. If you see people walking closer to each other than to other people at the mall, you tend to interpret that as being that those two parties are together, not as two separate individuals, because they are physically closer together. This is the principle of proximity. Similarity, things that appear alike, we group together. So when we look at the candy in the bowl, we tend to perceive those pieces of candy as being of the same kind. When we look at, um, oh gosh, let me think if I can, similarity principles. Oh, when you look outside, you can see different kinds of trees, different clumps of flowers, things that are closer together we perceive as being belonging to each other. So whether it's groups of people or groups of letters, um, that is proximity. Similarity, things that are alike. Um, we want to connect. So when you see the pattern of, think about the triangles and the circles, visual image that we looked at in class way back in the beginning of the year. The fact that we immediately, here we go, that we immediately perceive the triangles as belonging together and the circles as belong together rather than triangle, circle, triangle. We're going to give it some kind of meaning that they belong together. The principle of continuity. Continuity refers to the fact that we perceive, even though this is a series of broken half circles, we like to perceive the line as continuing. This is just one line, not several little half circles connected to a single line. Closure. Even though this is not a complete image, we like to fill it in. The principle of closure helps us to finish images so that we can interpret them faster. Connectedness. Connectedness refers to the fact that anytime something is connected, we believe it to belong together. So think about anytime you see a line drawn between things, we immediately presume that they belong together because they are connected. So proximity, similarity, continuity, closure, connectedness. These are all things that we are using in Gestalt psychology to interpret our environment. All right. So I'm going to wrap up here. We've only got a couple more things that I want to cover, some basic issues with perception and how we are giving meaning. Depth perception particularly is important to look at the example of the visual cliff. The visual cliff was in an experiment in which they put a young child who had just learned to crawl onto this table and then covered the table with plexiglass and put the same cloth down here underneath. And what they wanted to know is, given the option, would the baby crawl out over the glass, or would they stay back? Now, remember, this is a baby. They can't just look at that. Now, when you look at this picture, you can see that that's a piece of glass there. But this is a baby. They don't really know. So the question used to come to me, well, how do we know what the baby can and can't see? How do we know what the baby is thinking? Well, So the baby looks at the glass, and if you could see the bottom, you would see this same tablecloth, the same checkered cloth down here on the floor. So what we want to know is, can the baby perceive depth? Will they crawl out over the cliff or no? Now, obviously, we don't want to set this up so that there isn't something for them to crawl on, because otherwise, if they did decide to crawl, they'd go tumbling face first. But we don't want that to happen. So that's why the piece of plexiglass goes here. So baby crawls over to the edge, gets just about ready, and no matter how much mom calls the baby will not go out on the visual cliff. All right. Bear with me for a minute. All right. So now we're going to talk about a couple of other principles of perception. You want to make sure you're familiar with the ideas of constancy. Remember, constancy looks at shape constancy, size constancy, and brightness or light constancy. So basically what that tells us is that even with varying amounts of stimulus energy, we are able to continue to perceive the same thing as relatively unchanging. This enables us to accurately navigate our environment. So you see the fact that the amount of light being reflected off that paper is changing. That doesn't mean that the paper or that our perception of that paper is changing, but that even though there are several things that are different, for example, the amount of light reflected off of the paper, we are still able to continue with that standard, with that same perception. We don't have to adjust every time. Okay. So another issue is thinking about uh, shape con- shape constancy. Even though something is um, it appears to be different shapes, we can continue to perceive it. When the shape, when the image cast on your retina changes, we can continue to perceive that shape as having been the same. The very best example of shape constancy comes in an open and closed door. The closed door is way more rectangular and squared off. So it's a solid straight rectangle, where once that door gets opened, sometimes it doesn't cast the exact same image on your retina, but you're still able to perceive that same thing. That door opening and closing is still a door opening and closing, and it's in the same place, even though the retina image is different. Size constancy. We use size constancy so that we know that when we are standing here looking at this tree in our backyard, and then when I go out to the tree and I walk up to it, it looks a whole lot bigger. But the fact that I am able to continue to perceive that tree in about its same size, same shape, same coloring, this is all of our constancy cues. Which takes us on to monocular and binocular cues. Binocular cues are cues that require two eyes. We use both of our eyes to judge depth. One of these is retinal disparity. Retinal disparity is the fact that even though we have two retina that are each collecting an image, we are able to apply meaning to that and to bring it into one image. Our monocular cues are anything that occurs in only one eye at a time. You don't lose your depth perception with monocular cues. And let's talk about the different monocular cues that we want to be able to interpret. So we've got linear perspective, interpositioning, relative size, relative height, relative clarity, light and shadow, texture gradient, and the relative motion, also called the motion parallax. So let's talk about linear perspective. Linear perspective is that parallel lines appear to converge in the distance. We use these parallel lines to judge distance on the basis of the fact that as they appear to converge, we must presume that they are farther away. So these straight lines appear to converge or come together in the distance. Interpositioning is also sometimes called occlusion. Interpositioning is when one thing blocks something else. This has to do with the fact that the first object in blocking, it prevents us from getting the whole image of the second one. Interpositioning is in your textbook. It's not something that I would expect to necessarily see many, if any, questions on. Relative size. Relative size is a depth cue that causes us to perceive something as being farther away because it looks smaller than an object in the foreground, and we assume that therefore it must be similar in size, even though that's not true. As we perceive the people in the background as more distant and not as physically smaller, what we are doing is using these cues to determine how far away, how big, how threatening, etc. Relative height is a cue that makes objects that are higher in your field of vision appear farther away. If you draw a horizontal line across a picture halfway up, our brains assume that the top half is more distant and the lower half is closer. Relative clarity is the fact that a cue makes clear objects appear closer than blurry objects. Relative clarity has to do with the amount of detail, and this is looking at blurry objects, fuzzy objects. The farther something is from us, the less detail it conveys. This is the big issue, I guess is the word I'm looking for, in interpreting excuse me, I'm sorry. Relative clarity, back to the relative clarity. It makes the objects that are closer appear blurry or fuzzy. So think about airplane pilots. That's what I was trying to pull. Airplane pilots who are descending on a foggy day. Those foggy conditions will tend to misalign the pilot, and their actual perception of distance to the runway will be off. It'll have mistakes in it because of relative clarity, unless the co-pilot calls out the altitude throughout the descent of the airplane. Light and shadow. Nearby objects reflect more light than distant objects. So if there are two identical objects, the dimmer one seems farther away. Texture gradient combines several different monocular cues. Take, for example, look at the floor that you're standing on. It's true that some things will look significantly more textured. Why? Because we tend to find them closer. So when those objects are closer, we see more of the texture, whether it is, for example, an orange or the back of, there we go. The back of the piece of artwork that Cooper made. Oh, there we go. All right, so when you look at this, when it's farther away, you see less of the texture than when it is up close. So this is one of the cues that we are using to judge distance. Finally, the relative motion cue, which is also called motion parallax, makes the objects that are closer to us seem to move faster than those that are farther away. This is when you're riding in the car and your parents are driving and you look out the window. As you focus on things that are closer to you, they seem to be moving faster than those that are farther away. So let's move on. I think that gets us all of our big cues. You do absolutely want to know the Muller-Lyer illusion. We'll talk about those. I guess we'll talk about all the illusions in the next session. So I am going to sign off, and then I will post another with just optical illusions.